Okay, it's now 3 o'clock by my time, so I think we can get started. Welcome everyone to the LifePlan Publications webinar, where we will be telling you all about how to be a co-author and what publication plans we have for the remainder of our time together. So I'm going to briefly go over next year, the final push, and then explain how to be a co-author. And we'll give the award to the most helpful team, as is traditional. And then Otso and Tomas and the researchers going to take over and tell you about our actual publication plans. And at the end, hopefully, we'll have time to take questions from the Q&A, not the chat. And this is being recorded, and we'll be sharing it on the LifePlan website in the news section. So the current status of analysis, this is always on our status page in an updated form, this picture. We're now able to produce species lists or operational taxonomical unit lists for cyclone and soil samples, and some initial bioinformatics for malaise samples. Some local models are ready for the audio data, so we can get results for some places, but not all yet. And we're still putting together the annotation data for images, but we are progressing with the turning your data into species lists. Next year is the final year of LifePlan sampling, and we can now say that any samples that arrive to us by the end of October 2025, so arrive to Sweden or Canada by the end of October 2025, we will be able to process. Samples after that, we will definitely try to process them, but we can't absolutely promise. But do still continue until the end of 2025 if you can. We have multiple pending funding applications for at least continuing analysis and writing up beyond the project period. After the project period, we won't be able to financially support actual full LifePlan sampling as it has been going on. But we would be keen to contribute to analysis and writing up. But the point is that LifePlan is a method. It's a protocol that anybody can use. And some sites are saying that they are already planning to continue with the LifePlan protocol on their own funding after the project period. And of course, we're very keen to collaborate with that. So if you want help with analysis or writing, then of course, just contact us even after the project. And you do, of course, keep the equipment from LifePlan in whatever condition it's in, so you can keep going. And do remember that long-term time series are really rare and valuable. And the longer that you keep sampling in the same place with the same methods, the exponentially more valuable your data set does become. So do consider continuing beyond LifePlan if you can. Okay, which teams will be included in publications? What does it take to be included? Of course, you must have provided sufficient data. Your image and audio data must be uploaded to us, and you should have sent some physical samples in. Now, if you haven't sent any physical samples in yet, you are now officially in a hurry, because at some point we do have to cut it off and start producing papers, and it takes time from receiving samples. We have to clean them, we have to DNA sequence them. So there is a lag there. So if you haven't sent any physical samples in by January 31st, we really can't guarantee that you'll be included in all the publications because of that lead time. Your metadata must be checked. So once a year, we send you this map and list of where your traps are to check that your traps are where we think they are. So please do respond to those each year. And to find out whether your samples have been sent in, whether your image and audio data has been sent in, every month we send you a summary image that looks something like this. This is an example from one unnamed team. So you've got on the x-axis, you've got the weeks. You've got the years in these big chunks, 2021, 2, 3, 4. And we're at the natural site, urban site, natural site, urban site. And we've got the different sampling methods here: soil, malaise, cyclone, camera, and audio. Each of these little lines represents 1 week of sampling. And you can see the status of your samples by the color coding. So if they're in black, then they're still with you. You haven't uploaded them or sent them in. So please get on that, especially if some method is completely black here. If it's green, then you've sent it to us and you're fine. If it's blue, then we've already sent it onto the sequencing lab. It's in the sequencing queue, and if it's pink, then we have sequence data already back. Further, you must have your contracts in place. So your collaboration material transfer agreement, the agreements with us, those must be in place and signed. And you must have your other relevant permits in place depending on where you are. So some countries require all kinds of permits and some countries require no permits. So you are ultimately responsible for having those, and we want copies of the relevant permits sent to LIFEPLAN. Many of you already have, but if you haven't yet, then please do send those in. And how do you know if you've got your contracts in place? You can check our newsletter, which comes out every other month, usually. Your site should be in green or yellow here. But please remember that even if you are green here, there may still be further permits that are required by you locally. And I will be going around and asking teams that haven't sent permits in yet if I know that they're required. But they'll be required at the publication stage. All reputable journals will require permits, so it will be an excluding principle. Within the team, who will be co-authors? Every time we start the writing process for a new paper, I send a questionnaire out to the coordinators of the teams to ask who should be the co-authors for this paper and briefly explaining what the paper is about. For each team, you get a maximum of 5 co-authors, and the coordinator is the one who decides who they will be. The co-authors can be different for different papers. Maybe they should be. The people who collected malaise data might be different from the ones who collected audio data. But we do recommend that the co-authors should have been actually involved in collecting the data that are being published. What about your data and results? So you can ask us for your sequencing data if you see pink in your sampling summary. For bioinformatics data, when we do get it, I send that out as we get it. So you don't need to ask for that. And as you get your results, you can, of course, start working on your own analyses and publications as soon as you like. But please, again, don't publish your data or analyses until the joint LIFEPLAN data papers are submitted. And finally, from me, most helpful team for 2024 is Oulanka, based at Oulanka Research Station in Finland. They're getting the award for their consistent and just careful sampling, and most importantly, keeping their Nextcloud clean, which is very, very helpful, especially to me. So thank you, Oulanka. And now I'm going to hand it over to Otso.
Okay, thanks very much, Bess. So let me start by reminding that the aim of LIFEPLAN has really been to integrate statistical and ecological research. So we have these 2 legs, statistical research and empirical research. So in addition to this globally distributed sampling design that we have all put together and that you have been working very hard on, we have performed a lot of work to develop statistical methods that are needed to interpret these new kinds of data. And that's why this publication plan is split into 2 kinds of papers, so methodologically oriented papers and empirically oriented papers. So in the next slide, we just have an overview of all of these already published papers or submitted manuscripts or papers in preparation or papers in plans in terms of their numbers in these 2 categories, methodologically focused and empirically focused. And if you just sum up these numbers, you will see that there are actually 101 papers or manuscripts or plans in total. So there is a fairly large number of them, which is great, but it also means that it's not possible to go through in detail each of them individually in this webinar. So what we will do is to still give you the full list, so you will have them like in the screen in small font to see all of the plans that are going on, and so that you can then maybe from the recording, if you like, you can look at them in more detail. But in addition to these full lists, we will then highlight some of the papers that we think are maybe especially interesting in this context. And still, if we go back to the previous slide where we see the overview, what you see is that in these published and submitted categories, we have a lot of methodologically focused work, but not so much empirically focused work, whereas most of the empirically focused work is either in preparation or especially in plans. And this is, of course, the case because it's only now that we start to have like a really substantial amount of data, and we are ready to start with the empirically focused papers. So while we have been waiting for the papers, waiting for the data, we have been very busy in developing the methods, so that now most of the methods are kind of ready to go. Okay, but let's go to next slide. So we go in the same order as was in the previous slide, starting with the published methodologically focused papers. And I'm not going to read through all the papers here, but let's just look at the categories. So first we have the field sampling methods, and we are very happy that now this sampling protocol, LIFEPLAN sampling protocol, has been published or is in press at the moment in PLOS ONE. So that simply describes like how LIFEPLAN sampling is conducted, and I think that's a very useful reference for all of us. Then there is a very large number of statistically oriented papers, especially by the US team led by Professor David Dunson, which methods have been developed from the point of view that they would be then useful for the LIFEPLAN data now that we have the data available. And in the next slide, we see that in addition to kind of the statistics, we have conducted quite a lot of research in bioinformatics and machine learning to develop methods for species classification, especially for DNA data and audio data, how to identify which species are in audio and in DNA using automated methods. All right, so let's go to the next slide, which highlights one of these methods developments, and this will be presented by Dr. Gleb Tikhonov from the University of Helsinki.
Thank you. So here in the Finnish team, we have been working hard on accelerating our flagship model for analyzing species communities named HMSC, and which is formed as HMSC R package. So in the paper which came out last September, we proposed a novel approach not for the model itself, but to how to fit the model in practice. Basically, we proposed to port the computations for the model fitting algorithm from CPU to GPU, and we showed that such porting allowed us to achieve much higher computational speed up, which is very, very important in context of LIFEPLAN, because LIFEPLAN generates a lot of data, which previously would have taken tremendous time to analyze. We also have conducted a so-called porting program specifically for the computational resources which we have at hand, the Lumika supercomputer, in order to make this code run in the air as smooth and as efficient as possible. That's pretty much it from my side. Thanks.
Great, thanks a lot, Gleb. So let's go to the next slide. Moving to empirically focused published papers. And now, very strictly speaking, we don't yet have any published papers from the LIFEPLAN project. So we don't have any papers that are published from the data that has been generated specifically in this project. But we have recently published some papers from, let's say, a pilot studies before LIFEPLAN. So before LIFEPLAN, we had the global spore sampling project that many of you actually participated, and that was then kind of extended into the LIFEPLAN project. So we included here papers that have been published from similar data that we collect from LIFEPLAN, but mainly before the LIFEPLAN funding period. And I think these papers are important also from the point of view that they work partially as models of what kind of papers we can do with the LIFEPLAN data. So let's go to the highlights slide, which will be presented by Dr. Brendan Furneaux from the universities of Jyväskylä and Uppsala. Brendan, we cannot hear you.
How about now?
Yes, now we can.
Yes. Okay. So sorry about that. So this is a pair of papers based on the global spore sampling project, which is, as also mentioned, it's quite similar to the cyclone sampling part of what we're doing in LIFEPLAN, but it was collected a bit earlier. And one of the things to point out here is that it is a pair of papers, and this is also one of the things that we are planning on doing with the LIFEPLAN data. The paper that you see on the left here, published in Scientific Data, it just releases the data set and describes how we got the data and some very basic analysis, and then puts it out there for people to use in as useful a way as possible. And then on the right is our analysis of this data, or the first analysis paper on this data, where we actually go in and look at the seasonal dynamics of fungal spores that we find over the year of sampling that's represented there, and that's published in Nature. And we'll plan on doing a similar thing with LIFEPLAN, where we publish a data package and then do analysis and publish those analyses separately.
Okay, thanks very much, Brendan. So let's go to next slide. And now we are moving to submitted manuscripts. And again, we have quite a large number of statistical methods that have been submitted to journals. Many of these are actually already available in the bioRxiv or in the archive. And then we have the first work finished related to the camera trap data. So there is a species classification method manuscript led by Tommi Mononen from the Helsinki team, which has been submitted and is under revision. So let's go to the highlights, and this highlight will be presented by Professor David Dunson, who is the leader of the US team.
Hi, thanks, Otso. Yeah, so we've been interested a lot, motivated by the LIFEPLAN data on trying to scale up joint species distribution models, and particularly the HMSC type of approach for joint species distribution models. And one approach we developed was kind of motivated initially by the Malagasy arthropod data. So in these data, there's over 250,000 different presumed species or OTUs. And you can't just directly fit current HMSC or JSDM models. And so we came up with this common to rare transfer learning approach, where we would take the common species and we would fit an HMSC, and we would use that to extract latent covariates or latent factors of each sample, which are telling you a lot, or explaining a lot about the conditions in that sample. And it makes sense to then use those factors in a sort of a backbone model for the other species, use those factors to explain the dependence in the rare species. And so we use the common species to infer about the rare species. And we can do that also through a phylogenetically structured prior, or kind of hierarchical prior. And so you don't just like fit all of the species together. You use the common species to infer these habitat latent factors, and then you transfer that information to rare species analyses, also borrowing information through a phylogenetically structured prior. And so this worked really, really well. It's under review at Nature Methods, and we have another couple of methods in the pipeline that can do this type of scalability to hundreds of thousands or even millions of species, which is really, really important in analyzing the data that are now coming online. And I'll stop there.
Thanks very much, David. So let's go to the next slide, which is another highlight that will be presented by Dr. Tommi Mononen from the Helsinki team.
Okay, so this is a species detection paper. And the idea is to kind of, as your cameras take 5 images, the idea is to detect movement and kind of to find those animals from these images. As you see in the middle image, there is deer, and then the movement revealing panel on the right reveals that. So we can find animals behind vegetation. And yes, this method needs really little data. It's generalizable, and it also finds unknown animals, so animals it has not seen before. Thanks.
Thanks, Tommi. So then we move to the submitted empirically focused papers, and there is actually only one such manuscript, but this is a super important and exciting manuscript because this is like the first kind of a real LIFEPLAN product, so that this is like from the LIFEPLAN data, not the pilot project data before LIFEPLAN, and this manuscript will be presented as a highlight by Dr. Panu Somervuo from the Helsinki University. So let's go to the next slide.
Okay, so here we have utilized the data from over 100 sites and using more than 1 million 1-minute recordings. And we have like characterized the sounds by computing 15 different metrics. And on the left-hand side, that's kind of the table. Basically, it's just like they describe how the sound is distributed to different frequencies, and we have calculated commonly used soundscape indices, and then we have also like calculated some sound events, how they occur in our recordings, and then that alone like showed that this data is really useful. So we were able to find the patterns and rhythms in day and also in season. And one of the things we were like interested in is to compare those natural and urban sites, and that's the picture in the like middle, and some of which kind of, how to say, make sense results were that, okay, in urban habitats we do hear more those sounds which come from human species or vehicles, but then there were also some other maybe not that like expected results. For example, we were able to detect more animals and more birds in the urban sounds, and actually that also makes sense because that's no kind of Lombard effect. So in a noisy environment, many animals, also including humans, so tend to vocalize louder and also a little bit like higher pitch. So that's easier to detect. But this is really kind of the promising, and this was the starting point for the further analysis, and this kind of shows that our data is useful, and also the data which will be collected like later and allows more detailed analysis then. Thank you.
Okay, thanks very much, Panu. So with the next slide, we are moving to the future plans. So I hand over to Tomas, who will continue from here.
Thank you. And just one thing that I wanted to say is that at the start of the webinar, we didn't quite emphasize that we have this question and answer channel. You can't chat in the traditional sense, but you can send in questions, and those who aren't speaking will be trying to answer them. And also, if there is time at the end of the webinar, then we will pick questions from there and try to answer them together. So feel free to post questions through that channel. But with that said, I think in preparation and in thoughts section of our papers are really the most exciting, because as Otso was saying, I mean, now is the stage when we are when we have amassed these amazing global data, and we are all set for global analysis. So I think these sections will be really exciting. Naturally, we're not all done with the methods. There are methodological challenges left, And some of the papers will be focusing on that, also on evaluating the challenges and pitfalls of these kinds of high throughput data, and the statistical leg that Otso described will continue to be a very strong and active leg to stand on. Next slide. Now, with these massive data, very much of the work is focusing on and will be focusing on actually deriving species-level data from the indirect signs of species that we're finding in DNA and sound and image. And very much of the ongoing work is about how we kind of split the variation that we find into units matching species, how we evaluate the confidence that we have in assigning particular sequences or sounds to those categories, and then that actually applies to both DNA and sound and image. And there's work going on anything from arthropods to bats and mammals. Next slide, please. And as an example of something truly going on, I would ask Johanna to talk about a paper where we combine different modalities, so different types of information, to make more use of these data.
Yes. Thanks, Thomas. Yeah, so this is the paper where we're working on the data from the malaise trap samples. So as you know, the main data that we get from these samples are metabarcoding. But for a subset of samples, we also take these bulk images that you can see to the left. And the main aim of this project is to train a machine learning algorithm to count insects from these images, and also to classify them so that we can get abundance information per taxonomic groups. And as you might be able to see in the image example, it is difficult to classify insects from the image to lower taxonomic levels, such as species. But we do already have some of these classifications from the DNA sequences. So the idea is to use the sequences also to inform the classifier. And then to the right, you can see that we also have some individual specimen data for a subset of our samples that can be used to, for example, pre-train the algorithm so that it can make this connection between DNA and image. And in this project, we already have a working instance segmentation pipeline that can be used to count the insects. And we're now beginning the work on getting all these different data modalities together to create a classifier that's hopefully more powerful than from images alone.
Thank you, Johanna. And next slide, please. Okay, then we have empirically focused papers focusing on particular organism groups. Some of the most amazing ones will be about, well, I think Brenda will talk about some of those. We already have the data on, for instance, biomass in insects, the total biomass of insects across the world, which is something that Johanna is working on. And we will be applying some of the methods that we used in the Nature paper that Brendan just talked about to data on arthropods. Next slide, please. And this is actually the known and unknown fungi example that Brendan will elaborate on.
Hi. So this is another paper, again, based on this GSSP, although similar analysis will also be able to be done in LIFEPLAN for both the fungi and the arthropods. And so this is dealing with this issue that we have where most of the species of fungi are what we call dark taxa, which means that either they've never been described by science or we don't have a reference DNA sequence, so we can't link that name to a DNA sequence. And so then the question here is just what can we say about these fungi? Where do they occur taxonomically? So like what family or order should they belong to? But also what sort of geographical distribution do we see, and what sort of ecological distributions do they seem to have? And here in the graph, we can just see how this looks for fungi from that global spore sampling project. And we've got the different taxonomic groups going from left to right, phylum being the broadest, down to species, which is the most specific. And then just looking in different parts of the world, polar, continental, temperate, or tropical, subtropical, how good a job can we do at identifying those? And do we think that in the cases where we cannot identify them, we've got red, which is where we can't identify them because there's just too much uncertainty. We can't be sure what this is. Maybe it's something we know, but we can't tell for sure. And then this
Time,
blue.
Brendan, sorry.
Yeah.
Time.
Okay. And then the blue down at the bottom for things that we really are pretty sure are new.
Thank you, Brendan. And I just have to mention that Brendan delivered the first examples, comparative examples from animals from the malaise traps on Saturday, and they look partly contrasting. So this is really fascinating work. Next, please. And this is
Otso.
Otso.
Yeah, so what is really super exciting is that now we are talking about malaise trap data. We don't only have the samples, but the samples have been processed and sequenced, and beyond that, the bioinformatic pipeline has been done for this big set of samples. So now we have the data tables for more than 8,000 samples collected all around the world. And if you simply look at the species diversity, you see that it's really amazing. We have like 900,000 species level OTUs in this data. So now this is really one of the biggest systematically collected global data on arthropods that just came out from the Owen and is ready for analysis. So with this data, we will follow the same model that Brendan earlier presented for this GSSP data paper and nature paper combination. We will publish the data that is now illustrated in this slide here, which species we found and where each species was present as a data paper. But then maybe more interestingly, we will then make the scientific analysis of this data. If you go to the next slide, you will see something very fresh, because the data are very fresh, and these preliminary analyses are so fresh that I actually did them like 3 hours ago, just before this webinar. So they are not like yet the final results for the paper, but just to illustrate something that that the data has like a really exciting signal. On the panel on top, you see an example of seasonal dynamics of arthropod richness in a Swedish site. As expected, they peak somewhere like in the middle of the summer. Then I did fit these joint species distribution models separately for all of those 81 sites, for which there are at least 50 samples, so that you can really look at the seasonality. And I asked that how strong was the signal of seasonality in arthropod occurrence, not only in species richness, but in the community composition of those arthropods.
Time.
And then I simply plotted that here as a function of latitude to see that, in high latitudes, we have much higher seasonality than in low latitudes. Data has a clean signal. This is an exciting starting point for the analysis. Sorry for going over my time, but I just can't emphasize how excited I'm about the availability of this data.
Next, please. Great. Then we come to the part where we have only started the planning, but where we realize we will be able to do this as based on the amazing data that Otso was talking about. Much of this will again focus on actual methods, where we feel that based on both the insights and the new methods derived so far, we can actually reach even further, and there will be much further work on statistics. We will improve sampling methodology, and we will derive conceptual insights. as we're working all the time with these massive amounts of DNA, the effort going into bioinformatics is substantial, and there will be several papers. We're also working on improved image-based methods and annotated image resources to be shared again as open data. Next, please. And in terms of data on sound and images and the cyclone and the soil samples, again, we will be releasing these major data papers to the use of absolutely everyone, but also be using the data ourselves for analysis of truly global patterns in bird vocalization, in the co-vocalization between different species, both birds and bats later. The same for images. We will be releasing big data papers on that. And one interesting thing is that while we don't hold direct observations of species interactions, we do have information on organisms co-occurring in the samples. So we will be attempting to link, for instance, insect occurrence in the samples with changes in spore composition, thereby inferring how different insects carry the spores of different fungi. Next, please. Great. And then there will be work on the global phylogenetic structuring of communities, because that's part of the DNA information that we derive. There are plans for more regional analysis focused on, for instance, Madagascar. We're having intense sampling, and I think one of the most exciting types of papers are, again, the papers where we combine different data types to draw on all the different types of data to derive new ecological inference. Next, please. And here, Tommi has an example.
Yes, so this is a paper where we will look at the global data. So on the left, you see one site, and for example, species like yearly presence of them, curves there. But one side is not just enough. So on the right side, you see 75 cameras and over a year. So we have 75 cameras in Uppsala, and you can see much more, some general patterns there, like people, like animals are active on the sunset and sunrise, and birds, of course, in the middle of daytime. And also in winter, there are not so many birds. So we are going to look at this kind of phenomena over global data and latitudinal data. Thanks.
Thank you, Tommi. And next one, please. And here's another highlight presented by Patrik, please.
Hello. The purpose of this paper is to study the co-vocalization patterns in world bird communities. So the plan is that we will automatically identify the bird species vocalizing in the global soundscapes and then analyze the overlap of vocalization for all species pairs to find out if there are some species which avoid each other or some species which tend to overlap with each other more than what would be expected to happen by random. And we already have some initial results. So these two Finnish bird species, common chaffinch and willow warbler, seem to avoid full overlap of vocalizations, but it seems that they tend to respond to each other. So right after one species is vocalizing, the other species tends to respond to that. And next we plan to more carefully analyze this for all species in Finland and then repeat this study for global soundscapes and global bird species. Thank you.
Thank you, Patrik. Next slide, please. Great. And then I think the final highlight is one of the highlights of the full project, in fact, because remember that one of the main objectives of LIFEPLAN was to find out whether once we start mapping the formerly very dark taxa, whether they actually exhibit patterns which are comparable to the species that we know better from before, or whether they are structured in some entirely different ways, these communities. Because the background here is that most of what we think we know about biodiversity is based on the least diverse parts of that biodiversity, and many of the identifications of like hotspot regions and the regions of highest priority and highest endemism, etc., they are all based on vertebrates, which account for about 1 per mil of global species richness. And now when we get to data on groups like fungi and arthropods, where the true diversity is, we can expect, I think, to find entirely new patterns. There are some comparisons like these, but LIFEPLAN has generated the first fully standardized data set for making this kind of comparisons. And we will be looking at both global patterns in species richness. We will be comparing latitudinal trends, where again, initial indications are that the trends are very different in these species-rich groups. And we will be looking at patterns of species turnover of beta diversity. And remember that as a special feature in the LIFEPLAN design, we have this nested design to look at turnover over different spatial scales. And this we expect to be very, very powerful for this paper to come. And with that, I think we have ran you through our publication plans. I think we have flashed about 100 papers at you. We realized that you cannot possibly have gathered all the details, but what we wanted to show is how much we have already produced together and how much we will be producing before the project ends. And an important point is, of course, that LIFEPLAN doesn't end on the last day of March 2026 when our funding ends. I think that we will be focusing on these data for the next 5 to 10 years to come. There is so much to do, and there is also so much to build on from the methodological advances that we have already gained. So I think the final stage of LIFEPLAN will not be the end, but maybe the most intriguing phase of the project. And now we invited you to ask questions. Some of them have been answered, and some we could address together.
Yeah, I could take this one from Ika Jucic from Austria. So it has been mentioned that monthly status summaries are provided. Where can they be found? So each month I send the team coordinator this very annoying email that says, here are 5 Excel spreadsheets, one for each of your sampling methods. Please at least check this one. And for each row, it says, do something, do something. But there's also included in that email, there is this monthly summary picture, which is updated each month and sent. So if you go back, if the team coordinator goes back in their emails, they'll find their most recent one.
And I could take the next one, which is, my data has not been included in any of the paper. Is this a sign that there is a problem with the quality or handling? How do I know if there is a problem with my data other than the colors in the picture? Now, I don't know, because your name doesn't give it away where you're working, but I think we will tell you if there is a problem with the data, and we would not stay quiet and send you colorful graphs indicating that all is well, but kick you out of the paper. That's not possible. So I'm guessing, because the kind of key papers that have already been published, as Brendan was saying, they largely rely on data collected before LIFEPLAN in this global spore sampling project. And if you only joined LIFEPLAN when the real LIFEPLAN began, it's entirely possible that you didn't contribute to those data. So instead, you will then be included in the even larger papers to come. So I wouldn't worry if you are convinced that you have sent in the data.
Yeah, if I continue still from this, like just to clarify that out of these 101 manuscript papers that we mentioned, only one is such that it's actually based on LIFEPLAN data, and that is the Soundscape manuscript. So if you are not included in the Soundscape manuscript, then it appears that at the time that we submitted the manuscript, there was not sufficient amount of data from your site. Otherwise, you should be included in that manuscript, or then there is an error somewhere. But then all the other papers that we listed, like in the published and submitted category, they are mostly methodological papers which have been developed like without LIFEPLAN data, and that's why the authors of those papers do not include the sampling teams, with the exception that some of the papers have been developed in the context of kind of a pilot data from a couple of sites from which we had the data like ready, like the earliest. And then those teams are included in these method-oriented papers because they provided like fast pilot data that was needed to develop the methods. But now, as we presented in the plans, now we are really at the phase where we are focusing on the empirically oriented papers from the LIFEPLAN data, and then all the sampling teams will be co-authors in those papers if they, as Bess explained, like have submitted a sufficient amount of data and have those agreements ready.
And then there is while Bess is preparing to answer how you actually get the sequence data of fungal spore and insects, I will answer the one about the continuation of the project. As you know, 5 to 6 years isn't really a time series. Well, it is, and it would perhaps be very valuable to continue longer, at least on some aspects. And I mean, I couldn't agree more. 5 to 6 years is a very short time series. It is still a time series. We can do something with it. It would be amazing if we could continue at the same scale. At present, it's very hard to get two projects after each other at quite this scale. So our reasoning is that what we have done with LIFEPLAN is to show everyone how this can be done, to construct all the methods, well, almost all the methods for standardized sampling of biodiversity across the globe. And what we really hope is that these methods will simply be adopted by national agents, by continental agents, that they will partly be built into, for instance, European-wide monitoring schemes, yielding truly long-term time series. And on that part, we are working very hard together with the long-term sampling infrastructures existing in different places. And that's the kind of, that's basically the only way to secure the long-term uptake and survival of these methods, because there's nobody who's going to fund them for decades or centuries. It simply has to be taken up by these by these kind of new implementers.
Thanks, Thomas. And I can take the one about the sequence data of fungal spores and insects. So if you have samples that are in pink in your sampling summary, then that means that we have sequencing data for you. If you would like to have it, so this is raw sequencing data. It doesn't have bioinformatics done, so you need to be able to do bioinformatics yourself to get species identifications. But if you would like to have it, just email us at LIFEPLAN, and I will send it to you. Once we have produced our own bioinformatics, our species identifications, then those I will send to you as soon as we have them, so you don't have to specifically ask for those.
And can I just continue where I just ended, because now I see a message from Thomas Dirnböck saying, good news, the European research infrastructure ELTER has adopted the LIFEPLAN protocols. So all the European ELTER sites will therefore continue measuring after 2025. This is exactly the kind of uptake that we have been hoping for and which will secure the long-term survival.
And for Adrian, he's asking if it's possible to send in samples and data during 2026. So unfortunately, in 2026, no longer. This project ends at the end of March 2026, and then we can't pay our bills anymore, so then we can't sequence any more samples. So yeah, it's a 6-year project, but because of the long beginning of sending everybody their equipment and setting up the network, it's a maximum of 5 years of sampling in LIFEPLAN.
I could address the question that Alex Antonelli posted, which is about the process of assigning authors to particular papers. So is that by invitation only or a top-down process, or is it also a bottom-up proposal? And what should individuals willing to contribute to papers be prepared to do? And now this plan that we showed you, this 101 papers manuscripts, that's more or less the top-down part, that this is what we have been internally brainstorming what to do with the data, and what kind of publications we would like lead from the LIFEPLAN researcher team, and that we are kind of ready to make those analyses and write the papers. But of course, we are more than happy to go to a more bottom-up process as well. And I think there are at least two modes in which this can happen. One is what we are doing exactly now, so that if you have some ideas, proposals for papers that you think we should collectively do with these data, please post them now into the questions and answers. That that kind of a paper would be really exciting to do from your perspective, even if you don't think that you yourself necessarily want to kind of lead that work or have the skills to do it. But if you think that it's something that we should collectively do, please propose those kind of papers in the questions and answers now, or let us know later about those ideas. But then also, if you think that you would like to lead some analysis of these data, something that we have not thought about in our team and by our publication plan, and that you would have kind of the workforce and the expertise to do that, and in that sense, provide added value to the data, please contact us, and then we will discuss how that idea fits with the rest of the publication plans.
And I could actually take Alex Antonelli's other question about pushback on our neo-colonial approach. So we have actually had to respond to these kind of questions recently with the submission process in the Soundscapes paper. So I actually do have some text and material that I've put together. But I will. That's good to know that will be valuable to you as well. And I'll think about where to put that, if there's a place in our website to add that information.
And then I wanted to answer Thuania's question, where I think there was maybe a bit of a misunderstanding, because I think Bess said that you are not allowed to publish local data until the joint LIFEPLAN data papers are submitted. and that then you could only at this point publish acoustic data because that's the only joint paper coming out. I think Bess was a bit too strict about this, because my interpretation is that you are free to publish contributions on the local fauna as soon as you have the data, as long as you don't kind of team up with the 10 other sites within your continent or on the same planet to go for the kind of LIFEPLAN level findings. So unless Otso contradicts me, I would say local papers targeting data from a single site, you are free to publish as soon as you have the data in hand. And that is also one reason why we send you the data or make it available to you up to half a year before, for instance, releasing DNA data or such to others. So you can actually work on it. Isn't this the general policy? Otso looks happy enough, and Bess is nodding.
Yeah, Bess is nodding, so then that is indeed the case. Hey, I could address this question by Stephanie Boyer, which is a question about co-occurrence, where we mentioned insects co-occurring with spores that they may be carrying. And then the question is that will you also look into parasites and parasitoids in the same way, so co-occurrence between parasites and their hosts? And I think the answer to this question is very much yes, that many of these parasitoids, they will be revealed by the COI metabarcode that we will also look at use for their hosts. And in that sense, yes, we will look at the co-occurrence of the host and the parasitoids. Of course, we will not have for most of the data the resolution to say like exactly which parasitoid or parasite was inside which host species, but at the level of these bulk samples, we will be very much looking at this. And then as a bit similar kind of a species interaction from co-occurrence question, it's also possible to look at some bacteria from COI data, for example, Wolbachia, and we have already did some preliminary looks at that aspect of the data to realize that yes, this malaise trap data has very much signal on Wolbachia distributions and diversity globally, and that's something that we can then also relate to the variation in the arthropods.
And we are soon out of time, but I will pick the question from Scott Miller at the Smithsonian, who is asking whether we could incorporate additional data from the global malaise trap program, as they are indeed actually collected by the same type of traps, and we modeled very much on our protocol on that very initiative. And I think that's a great idea. Some parts of the processing are different because very much of the global malaise trap data are based on picking out the individual insects and doing individual level sequencing, yielding superb, for instance, quantification as compared to the bulk samples that we collect in LIFEPLAN. I do think that combining these two data sources for addressing questions about global species richness and global species endemism, etc., that is a very, very promising initiative, and I'm pretty sure we will do that.
And I could just take one final question from Jorge del Angel. So he's asking about samples that haven't been sent in, because I know in lots of places there's a lot of problems with export permits. If you get your samples into us before October 2025, we will promise to process them. The issue with being included in all papers, that might be a bit risky if you haven't been able to send samples yet. For instance, in Soundscapes, not quite all teams were included because some had not uploaded data by the time that we had to actually submit and cut off. So there is a slight risk that you'll miss out on some papers, but you will get results as long as you send before October 2025.
And before closing the data input to a paper, we have been very constructive in reaching out to the teams and say, do you want to be included? Then you really need to send the data, and then we define a date by which you need to send in the data. So it's not done without pre-warning. Just do, so you all know what is happening.
And I think with that, we are out of time. So thank you everyone for joining us today. We had a really nice participation, about 80 people listening to the webinar, and thank you for all your questions. And we'll be in touch with the LIFEPLAN email. Thank you. Bye-bye.